Hello everybody, and thank you for coming to listen to my presentation. Today, I'm going to be discussing with you my research at Nationwide Children's Hospital Institute for Genomic Medicine regarding the confirmation of somatic variants at ultra-low frequencies in seizure disorders using targeted amplicon sequencing methods. First, I will cover some background regarding epilepsy and the epilepsy protocol at Nationwide Children's Hospital. Epilepsy is a common disorder that is characterized by seizures. 20 to 30% of cases are acquired, such as through a stroke or brain injury, but most cases have a genetic etiology and arise through something known as somatic mosaicism, which I will cover in the following slides. At Children's, the epilepsy protocol is as follows. The patient undergoes surgery where a section of their brain is resected by neurosurgeons. The Institute for Genomic Medicine, or IGM, extracts RNA and DNA from the sample in the blood of the patient to use as a normal comparative. Next, the samples are sequenced using next-generation sequencing technology. The data is then analyzed and somatic variants are identified. Somatic variants will exist in the brain but not in the normal DNA of the patient's blood. Somatic variants that are called but have low supporting reads will move on to the next step, which is targeted sequencing, a process that I will also explain in the coming slides. Earlier, I mentioned somatic mosaicism. Somatic mosaicism occurs when a mutation arises in a cell lineage, as indicated by the graphic. As a result of this, a collection of cells will contain the mutation, but the rest of the cell lineage will not. Somatic mosaicism in the brain can lead to certain conditions, epilepsy being one of them. One common pathway that is mutated in epilepsy cases is the mTOR pathway. A pathway is a cascade of proteins that eventually leads to a certain function. As you can see in the mTOR pathway, there are many places wherein a mutation can cause epilepsy. mTOR is one of the mutated genes that was validated in a patient at NCH by targeted sequencing. Now, I will discuss the experimental design for the targeted sequencing that I have mentioned. First, a PCR primer set is designed so that the variant of interest can be targeted. This involves choosing a 250 base pair region that contains the variant. PCR will amplify this region millions of times. Next, an NGS library is prepared so that the sample can be sequenced on Illumina's iSeq or MiniSeq. When the data is returned, the variants will be analyzed. Targeted sequencing produces many more reads than typical whole exome or whole genome sequencing. This is because targeted sequencing focuses on only about 250 base pairs, whereas whole exome sequencing must sequence thousands and thousands of base pairs. Therefore, a variant that exists at ultra-low frequencies can now be validated due to the sheer amount of supporting data that can be provided from targeted sequencing. The applications of targeted sequencing are very exciting. Not only are we able to ensure that a variant detected by whole exome sequencing is real, but we can determine the tissue-specific prevalence of a variant by identifying the resected tissue type. Alongside this, mosaic variants that are present at ultra-low frequencies are able to be identified. This means that a very small percentage of cells in the sample can have the variant and it can still be validated. In the future, IGM is hoping to pursue single-cell experiments. Through single-cell, specific cell types that contain the mutation can be identified. This slide will show you the variants that have either been validated or refuted using targeted sequencing at IGM. The genes in the second column were identified as containing a variant through whole exome sequencing before targeted sequencing occurred. The furthest right column shows if each variant was validated or refuted. As you can see, from the 10 patients and 20 variants, 11 variants were validated. Here is a visualization of some of the variants that were either refuted or validated. Each bar represents a different type of tissue. The bars are separated by subject in the respective gene of interest. The blood should contain 0% of the mutation, otherwise it would not be considered somatic. As you can see, variants such as the one in Atom 22 and Subject 22 are clearly real, existing at 15.6% in the tissue. Sample C from Subject 23, shown on the right side of the graph, had a variant at a frequency of only 0.3%, but we were still able to validate it, even at that ultra-low frequency, thanks to targeted sequencing. You might be wondering why Subject 19, with a frequency as high as 1.1%, was not validated. This is because the variant was present in the blood, suggesting to us that the region was hard for the technology to sequence. Through this, one can see the importance of validating variants through targeted sequencing. Some variants that may seem real in whole exome sequencing can actually prove to be a result from sequencing error. Alongside this, variants that would be nearly impossible to validate from whole exome sequencing due to a low amount of supporting data can be easily validated through targeted sequencing. It is a relatively quick and inexpensive method that is helpful in determining the validity of a detected variant. I would like to thank Nationwide Children's and the Institute for Genomic Medicine for all of their wonderful work. Alongside of this, thank you to those that have listened. Have a good day and stay healthy.